Welcome back, guys. As we reconvene, Dr. Nikhil Patkar will take us through the techniques of library preparation and NGS panel designing. Let's welcome Dr. Patkar once again.
Hi everyone. This is a more technical talk on how to design a panel for NGS and library prep. So what most people do when they want to design panel, what they do? Call company wala. Okay, एक NGS वाला panel बनवा दे. So he'll say solid वाला बनवा दो, heme वाला बनवा दो, heme वाला बनवा दे. So that is the what most people do. When we were new to this, even we did the same. So or you take whatever company offers. So that time it was Illumina, TrueSight, Myeloid panel. So just buy that. So I want to change that, and I want to introduce you to the concept of how panels are actually designed, how to go about it from scratch. Okay. So first thing that we need to know is what workflow you want to choose. So this is scientific discussion, but is also economical discussion. We are not America, so we don't necessarily have all the resources in the world where we can say, like if you went to St. Jude, every patient gets 200x whole genome transcriptome sequencing. They don't care how many samples go on the machine, whether you are breaking even or not. You have to give result in like a week or 10 days and do whole genome, whole transcriptome. So we don't necessarily have that kind of funding. We don't, if it is there, it is only in some research project or something. But to deliver this routinely at a scale that you want, with the speed that you want, requires you to also have an understanding of economics. What kind of, your patients would be, what kind of payments will they have? Are they going to be like us, where money is tight? Or there you are going to deal with private patients, but even then, even in private patients, even private companies, there is lot of competition for delivering the cheapest test, to get more money. So based on that, these questions need to be asked. Where we will go eventually one day is whole genome sequencing, wherein you can detect everything in one assay. You can detect most of the CNVs, translocations, mutations. The disadvantage is that it has a very high scale of data. The whole genome, just if you do on a regular server, the mapping can take anywhere between 5 days to 7 days. Just the mapping of the genome, because that scale of data is so huge. And still it is low coverage. Everybody says that the $200 genome is here. It is here, sure, but it is 20x Mendelian genome at 200x. And you still need to buy a $1 million NovoSeq X and fill that flow cell, which costs like 22 lakhs for you to get that $200 whole genome. So there are lots of pros and cons in this. It is, as of now, not suitable for clinical genomics testing. This field will change. Exome sequencing, you capture all the coding regions of the genome. So anything that codes for a gene you take. You have exome sequencing, which is clinical exome, which is a focused 500 gene panel. You have actual exome sequencing, which is all the coding regions of the panel. You have exome plus UTR, which will do coding regions plus untranslated regions of the genome. And all of these are sold as different kits. Make no mistake about it, exome sequencing is also targeted sequencing. The workflow that you are going to do hands-on prep is entirely the same for exome sequencing as it is here. Okay, it is very, very well suited for Mendelian disorders. So inherited disorders, germline disorders, you have a trio, you have a proband, and you have you have a amniocentesis or something like that. You want to find out the genetic cause of it, you do exome sequencing. But it is suited for Mendelian, it is not suited for exome, because even when you do exome sequencing, it is cheap now. You can do an exome sequencing anywhere between 15 to 20,000 rupees depending on what instrument and so on. Even cheaper. If you have your own everything, you can do it at 10,000 rupees if you have a large enough sequencer. But you will still be able to get only 50 or 100x coverage. It is not suited for your somatic disease. There are other applications to somatic. Some people, what they do is they have a panel, but they will spike a exome in that. you can at a lower ratio, so that it will do everything, but you will get high coverage of what genes you want. You can do an RNA sequencing with an exome spike in so as to enrich these regions. Different sort of tricks are there. What is most commonly used in the market are DNA panels. They are used for cancer diagnostics worldwide. They enable focused interrogation, high depth of coverage. The caveat here is that if your panel is designed incorrectly, you can miss mutation. So in 2021, we added UBA1 mutation. Now we have 4 or 5 patients with Vexas syndrome. So if you are aware of this, you will get this. If your panel didn't have that, you will not have these kind of things. Transcriptome sequencing well suited for detecting fusions and expression abnormalities. You can detect mutations as well if you do whole transcriptome sequencing. Coverage may be higher, but it is, transcriptome sequencing, depending, this statement is kind of old, where it is unfeasible for clinical genomics testing. As of today, as sequencing costs drop, we may shift to an whole transcriptome sequencing altogether, but it still takes time. And bioinformatic, just the processing of a pipeline of a single sample for whole transcriptome sequencing still takes 1 to 1.5 days. Even in St. Jude's, we are collaborating with them on some projects. They also said it takes that time, because that's the time it takes for the pipelines to get through. Targeted panels can also be made on RNA, where you look at fusions involving where the goal is to detect fusions, and especially these are partner agnostic fusions. So I will talk about what they are, and these are now rapidly becoming a replacement for fish ashes, and they can detect translocations, but only from a panel. If something is new, you cannot detect it. So how do you design a gene panel? Don't call company wala. Do your own homework. So how to do this? Review the literature thoroughly. So what do you mean by review of literature? If you're doing let's say you want to do a myeloid panel, look at all the common myeloid disorders, download all those papers. In every paper, there will be a supplementary data. Okay. So in the supplementary data, there will always be a gene, the gene symbol. It will have the NMID that was used and what exons that there are. Most sensible reviewers will not accept a paper without this information, and that constitutes your panel. This is what you need to look at. Okay, you want to see what genes you want, and you want to see what published literature are there for them. Then you have to look at large repositories for mutation types. So as the genome project advanced, we similarly had you had more studies which looked at different ways in which cancer knowledge could be shared. The largest amongst them was the TCGA Atlas. All of you would have heard of TCGA Atlas. It's known as the Cancer Genome Atlas. It is a study across the world for common cancer types. Do you know India is also a participant of the TCGA Atlas? We participated in the head neck cancer project. So totally led by Dr. Partha Muzumdar at NIBMG Kalyani and by Tata Memorial Center. I think at that time Dr. Rajiv Sarin led it. Alright. So look at these repositories. These repositories are maintained, the TCGA repository is maintained online free of charge by Sloan Kettering, and it's called as cbioportal.org. Then St. Jude's has done lot of pioneering work on pediatric cancers, right? So anything in pediatric cancers you want to look at, please go to pecan.stjude.cloud. This Pecan is an absolutely amazing website that is maintained for them. And lastly, the biggest repository or anything you want to look at cancer is you go to sangerinstitute.uk. Now if you look at this, these are all the same guys who were part of the Human Genome Project, because they had that expertise, they had the knowledge, and they leveraged it to different kinds of projects. They had a head start. In 2001, I don't know, I just finished med school. I was not doing anything relevant, but I hadn't even heard of these things then, but they had finished the whole genome at that time. Alright, so now this is how the cBioportal looks like. So if I want to design a myeloid panel, there will be a thing here which will say all the different genes are there. I've selected myeloid here, and then in that myeloid it will tell you what are all the different myeloproliferative neoplasms, MDS, histiocytic disorders, AML, and this. So anybody knows what this is? These are all seminal papers that have been published in the field of AML. So if you see this NEJM 2013, this is the TCGA paper. If you see this one, if you see this is the OSHU paper again on cancer, on AML phenotypes and transcriptomes and epigenomes and therapy. This is the target initiative by Sohail Michinshi. This is all these are legacy studies that have been deposited. If you click on each one of them, you can click multiple one of them, it will highlight all the kinds of mutations. This is the first paper that was published from Japan, which showed that there are recurrent RNA spliceosome gene mutations in MDS. Today, everybody who's even in second MBBS will tell you that SF3B1 gene is mutated in MDS. So this is the origin of all of these papers, and they are all deposited on this website. Once you click the website, it will you can see all of these different genes are there, the mutations are there, and if you click on mutations, it will tell you what are the different mutations that are there and what is the frequency that is there. So now you know that in AML, what are the top genes that are mutated, and logically it makes that TET2, DNMT3A, ASXL1 will be common because older people will have more of those mutations, NPM1, SF3B1, SR, SF2 and so on, and you will see all of these. There are of course these statistical nonsense like MAP2K, but only 6 patients, you can straight away exclude this. So you can download each and every one of this data like this. So you select what you want and you download this data in a TSV format. Alright, so now whatever is there on the website, you can download into your format. The second resource that is extremely useful is Cosmic. So Cosmic is a very, very wonderful database. I cannot emphasize how useful it is for all the routine reporting. Alright, hematolymphoid tissue, include all sites, hematopoietic neoplasm, AML. Export what you see as a CSV file. CSV is comma separated values. Immediately you get that data on Excel and you can see all the different mutations that there are. Now any cancer in the world, any cancer, okay, if you look at the distribution of mutations in that cancer, you will get this long tail. Long tail of mutations means some one mutation came in some one gene, it will be represented here. But it's out of 5,000 patients, some three dots will be there. Do you want to get that? So this is known as the long tail of mutations. Why this is important? Because any panel you design, you have to address the problem of breadth versus depth. If you have a 300 gene panel, sure, chances are you will find 99% mutations in AML, but you are going to sequence 200 of those genes unnecessarily. Okay, because of that one mutation you want to see. So you have to do this balancing act between breadth and depth, where you have to decide how much wide your panel needs to be versus what you can afford. So we have a 135 gene myeloid panel, which we think is apt. We made a mistake initially when we got a Nexic, we went all out, we made a 225 gene panel. It had all kinds of Fanconi genes, this, that. We found one Fanconi anemia in 2 years. At the end of it, we realized this is nonsense. We are just wasting money unnecessarily sequencing all those. We are better off doing an exome in those patients anyway. So we cut our panel down to now 135 genes and we balance breadth versus depth, wherein we can reasonably manage cost and so on. So you have to say that I will cut off all my mutations at, let's say, 1% cutoff or 2% cutoff, and anything over 2% I am going to take. So you have to do a balancing act between breadth and feasibility, largely to maintain turnaround time. Now you have a gene set. You know that, okay, I am looking at this exon, this I am looking at these genes in AML. But how do you know which exons you have to choose, and how to know what are genomic coordinates? So now let us see what is happening. This is a site which is known as UCSC Genome Browser. So UCSC Genome Browser is a graphical display of all the cancer genomes that are there. And all I did, I went there and typed TP53. Now you see what is happening here. This is one TP53. All of these are TP53 genes, and there are all so many of TP53. So it is very confusing. So why it is necessary to confuse us by having so many gene versions. Alright, so a gene or a genomic region producing is a genomic region which when it gets transcribed produces produces mRNAs that encode a protein. a transcript can refer to any RNA that is produced from transcription of a genomic DNA sequence. And one gene can have many different transcripts. So all these lines that you saw are actually all the different types of transcripts that are active for TP53. So you remember when we said in the structure region of the genome that although our breakfast cereal has more number of genes or double the number of genes as compared to a human, we know how to use the genes better, and this is by extensive gene regulation, gene splicing and control. So this is what it means that so many. Do you know that in the that elephants never get cancer? Have you heard of this? Okay, elephants never get cancer because they have multiple copies of the TP53 genome. Okay, and that TP53 prevents any cancer that from being regulated. So clearly there is more to genes and gene regulation than meets the eye. So these genes can have different transcripts or isoforms, and separate transcripts may overlap in any given position in the genome. In one particular tissue, one transcript may be more active. In another tissue, the thing can be less active. We still don't understand much of it. At least I don't. Okay. So protein coding sequences in the genome are summarized in the set of transcripts we believe to exist, and variant annotation depends on these transcripts. Therefore, you have to understand what these transcripts are. So, National Center for Biotechnology or NCBI reference sequence is known as the RefSeq project. It provides records for any different organisms. They can be bugs, they can be bacteria, they can be viruses, whatever, or humans, and human genes. Provides sequence records for numerous organisms and provides a baseline for medical, functional and comparative studies. All of these are characterized by an accession format. Okay, so the accession format is 2 characters followed by an underscore. So if you see, you have heard of NMID. Most of you would have heard of NMID and genes. So NMID is like this: NM, that is protein coding transcript, underscore followed by an address, which is NMID. You have a NGID, you have an NCID, you have various other IDs that are there, NPID. What you require to know, I think, is NPID, NMID and like NCID. These are important, and this is an initiative of the US. It is freely available, but these things you need to know. The second thing that you need to know is a European initiative called as the Ensemble Genome Project. Alright, so the Ensemble project is also an extremely useful web server for you to get genomic information. So now let us see how we will use information. So suppose I am looking at TP53. I click TP53, I go here and I say, alright, show me all the different transcripts that are there. So I went to gene allele. So what you can see, the minute I type TP53, I get so many different transcripts. So it says that yes, TP53 is here and there are number of transcripts of the TP53 which are active. So it is lot of craziness, because if you all know of mutations, right? So we all say that IDH1 R132C, or we say IDH2 R140Q. But means what? It is with reference to one particular transcript, and what one transcript says in one lab may not mean the same as another lab. This has serious implications, not so much in cancer, but especially in the field of chorionic villus biopsy testing. There have been instances where one lab has said given mutation nomenclature, and the other lab looked at that position and said it's not there, and they said the pregnancy should not be terminated, but then the fetus was born with this abnormality. This is especially true if any of you has worked in thalassemia literature. Do you know of IVS 125G2C? Everybody knows it's the commonest thalassemia mutation in India. But what is this nomenclature? What is IVS 125G2C? This is legacy nomenclature. Okay? So this is not to be used at all. In today's time and age, you must follow HGVS nomenclature, and there is a joint incentive that was taken between NCBI and the ensemble, which is known as the matched annotation from NCBI and ensemble, called as the main project. Okay, and whenever you are looking for a transcript or you are saying that XYZ gene is there, what to look for? Look for the main transcript. The main transcript will save you lot of trouble. I will tell you. These define a high value set of transcripts and corresponding proteins. The main transcript are universal standards for variant reporting and browser display. So now that I went to ensemble, first thing to note is what genome ID is there. Okay, so we all described GRCH37, GRCH38. Make sure this is right. Suddenly you will find that your browser page refreshed, or you changed your computer, you went to office and then did what, or you came home and tried to finish what you are doing. You didn't pay attention to this. You are finished. Finished means you will put something and then something else will get synthesized. You will end up reporting this is always negative. Then you will see, अरे पता है Indian data में ना IDH2 आता ही नहीं है. Then the reason is इसलिए नहीं आता है क्योंकि तुम्हारा genome coordinate गलत था. Alright, so this is the transcript ID. You note the transcript ID, and most importantly, note the ref-seq match. So this is the address that you are trying to follow. Alright, you are see that this is a main transcript. You are seeing that this is a canonical ID. Canonical ka matlab that this is what has been used hierarchically. There have been more transcripts of IDH2 that may have been described. Doesn't matter. We are going to use the canonical ID, the one that is used most commonly in literature. All right. And so main select is great. Most of the genes have main select. Some will not have. In that case, I will tell you what to do. Look at the strand orientation carefully. So there are genes which are on the positive strand, and there are genes encoded on the negative strand. That means some genes will be counted 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 exons, and some genes will be counted 10, 9, 8, 7, 6, 5, 4, 3, 2, 1 because they are on the minus strand. The two strands of the DNA, they are on the reverse strand. Alright. So that is important when you are doing exon nomenclature, because you will take first exon thinking it is exon 1, arey but nahi, ye to exon 10 hai. So you can make a mistake there. Most importantly, don't ask company wala. Remember, this is the guy who is going to bring all his bias to the table. He is doing what some other company wala senior told him, and he does not know anything more from you. If you put little effort, you will be more knowledgeable than him for sure. What? It will be there in the orientation. It is. You don't have to change anything. If it is there in the reverse strand, it will be told like that. -7 means they may have noted it like that, but the transcripts will change. You have to be careful that you note that accordingly. All right. So now this is NMID. You can see that different things are there. Now let us say you don't want to get full transcript. Okay, so you don't want to get like IDH2. Anybody knows IDH2 exon 1 mutation, exon 2, exon 3 mutation, 6, 7, 8, 9? No, only thing you are interested in is IDH2 exon 4 mutation, right? For hematology, for any hematological malignancy, IDH1 and 2 only occurs in exon 4. This is the canonical area where mutations occur, and you want to take only this exon. You are not interested in everything else. So how to take coordinates from one exon? So this is what I need to look at. First I go to Cosmic and I see the distribution of these mutations, and everything is there only in this and this. So we know that this is exon 4. But how do I decide this is exon 4? Again, go to Ensemble. Go to Configure Options. Say Show Exons Only. Don't hide variants by frequency. Show exons only, intron basis वगैरह ये सब note करो. And then you come here. Okay. Now this page is going to show entire exonic sequence. It is going to show this number 1 here resembles the first exon. Chromosome start, chromosome stop, the length of the particular sequence, and you have to type download what you see. So when you download what you see, you get this. All right. So now each of these resembles an exon from 1 to 11, and you are interested in exon 4, and it is telling you that it has 161 amino acids, and these are the sequences that you are interested in doing. What is phasing? Phasing means that the boundary falls between two exons. Let us say that a codon has 3 bases, right? And ATG codes for methionine. Now between exon 1 and 2, this methionine is there, where the A lies, A and the T lie, or the A lies in exon 1, then there is an intron, and the T and G lie in the exon 2. When the thing is transcribed into RNA, ATG comes together, but this exon was broken apart. So it is going to tell you the start phase and the end phase, and this is what the phasing refers to. That 2 means 2 bases were here for the first thing, and then one fellow is here, and 1 means otherwise. You can look at it and you will logically understand what is going on. Alright. Exons have start and end phase, whereas introns have only one phase. A boundary in a non-coding region has a phase of -1. Now that you do this, you can download this, or you can copy this into a text file. These text files are the genome coordinates for that particular exon, and they are represented in the bed format: chromosome, start, stop, and comment. Comment can be anything. You can put your own name, but that would be counterproductive. Better to put exon and the gene name. Alright. So some of you asked, what is this minus and first? Now you see the numbers that there are. The stop number is 747 and the start number is 587. Aisa kaisa? Because it is in a reverse strand. It is counted backwards. Alright. So it is important to note this, especially for amplicon based assays. For example, thermo or Chi-seq or anything else, because you will get ulta orientation of primers. Alright. So it has importance, especially when you want to, when your assay may be looking at different things differently. So mandatory information first 3, rest is optional. Alright. So then what you do, is this okay by some final okay? No, you make everything on your text file, you copy this information and double verify it. How to double verify? Go back to UCSC genome browser, paste these first 3 coordinates with a colon and a dash, and it will tell you that yes, it is going to IDH exon 2. Now you know it is right. Because if you hover over this IDH2, it will show you exon 4. So now you know that your genome coordinates are right. This then goes into your panel. Alright. So a collection of these makes a gene panel. This is now given to whichever company in the world you want your gene panel from. It can be Twist, it can be QIAGEN, Illumina, Pillar, or it can be you yourself. You can also download, you can design your own primers, you can do your own assay, whatever you want you can do with these genome coordinates. You can say that I want to design only an assay for IDH R140Q, you download these coordinates, you use this, you basically run this through the genome to bed tools or something, and you can get all these bases out. And you can put this in primer 3 and use it to design an NGS assay. You can do that as well. But typically you want to do this for hundreds of genes with possibly thousands of exons, and it's better to use an bioinformatic process rather than to do it ad hoc. Now, how to decide how to prepare your library? Library preparation is a process of preparing DNA for use on a sequencer. Okay, this is how you prepare your sample for use on a sequencer. The term library refers to fragments of DNA with flanking components which enable it to be ready for sequencing. The size of the DNA is between the components and it's referred to as insert size. Library preparation is central to any sequencing process. It doesn't matter what genome sequencer you have, even if you have something known or unknown, you will still need to prepare a library. You have DNA. If you have a short read sequencer, Illumina, BGI, Ion Torrent, Avity, PacBio also make short read sequencers. They are called as the ONSO. Alright, you need to chop your DNA into small bits. That can be done by 3 processes. Number 1, you can use enzymes such as DNase 1 to chop your DNA into small bits. You can do sonication, which requires you to buy an expensive instrument called as a sonicator, which will do mechanical shearing of DNA by USG or by ultrasound waves. Or you can just do a PCR of 150 bases exactly to make short fragment DNA. So you get DNA of the size you want. 200 to 400 bases is what you're looking for. Then you ligate the instrument adapters to it. Okay. Now these adapters are specific to that instrument. So Illumina has their own, BGI, Ion Torrent, Avity, everything has their own instruments. If you have a long read sequencer, you are trying to get the largest config possible and you don't want to have any shedding. So if you are specifically looking for Oxford Nanopore, if you are going to do Promethean sequencing or something where you are looking at large deletions, then you don't need any of this. You just need to take the largest fragment of DNA. Even then you have to give careful thought to how you are going to extract your DNA. Or if you are going to use the PacBio Review, which is their huge NovoSeq scale machine where you want to get large regions of the genome, like a T2T project, you have to do special ways of DNA extraction and then you ligate these specific adapters to it. There is a third way to do this, and that is by a very proprietary enzyme called as transposase. So Illumina has this kit called Nextera. Have you all heard of it? Nextera kit is transposase based. Now many other companies also have Nextera. Nextera is a transposase based enzyme. The reason, the way it works is it recognizes particular fragment repetitive segments of the genome, and if they are there, it chops it up. If they are there, it chops it up. So this is called as transposase based library prep. You add your adapters into the transposase and in a single step you can incorporate these adapters, and you either PCR or PCR free you can get these adapters. So this is expensive, but yes, it is a way of doing PCR free library prep. Alright, now how do you get your template to your desired length, and most importantly, what is this so-called desired length? So the length of the DNA strand is obviously dependent on extraction technique and what has been done to DNA. If you take formalin fixed tissue, obviously you are not going to get 500 base pair amplicons, or if you take ancient DNA, you are not going to get, but in fresh sample you are going to get. Alright, short read sequencers typically require insert between 100 to 150 kb. Let us take a look at example of Illumina and Thermo Fisher. So this is straight from their website or something, I don't remember, article maybe, but it is openly available information. So if you use iSeq, MiniSeq, MySeq, NextSeq, almost all of them will have 2 into 150 bases. Okay, all of them will have 2 into 150 minimum. So what is this 2 into 150? Like I said, paired end sequencing. That means one read of one read is 150 bases this way and 150 bases in another way. This allows you to be slightly over smart also. You can make 300 base pair amplicon and get it sequenced on any of these instruments. Okay, so you can do 300 base pair amplicon, provided it slightly overlaps, it will recognize this, and when you do paired end assembly, you can make 300 base pair amplicon. 2 into 300 means on the MySeq you can sequence 600 bases. This is useful. Useful when you want to do IGVH sequencing, keeping leader based on leader, the primers are based on leader sequences. Or if you want to do let's say metagenomics, where you want the full length of the V1 to V9 gene, or you are trying to do FLT3 ITD detection, which is very accurate. It is largely dependent on read length. Even there, a large a large read length machine is important. Similarly, for NovoSeq, you can do up to 500 bases. For the Thermo Fisher, it is 600. But remember, there is no 2 into, because this is single end, not a paired end sequencer. So know your experimentation, know your flow cell, and that makes experimentation easier. If you talk to company wala, he'll tell you the most expensive product. You don't need 2 into 300 bases. This will do just fine. Okay. So how do you do this? Cell-free DNA, FFP DNA typically require minimum fragmentation. Large fragments rarely exist, but they are all require minimum or no fragmentation. Fragmentation is done by 3 ways. Number 1, sonication, which used to be done olden times or earlier, few years ago. Sonication was very popular. It can also be done by using enzymes. All enzymes, fragmentation enzymes, mostly use DNase 1. DNA comes from bovine pancreas. It's a powder from bovine pancreas, which you can get commercially very cheap, or you can get an recombinant enzyme called DNase 1 and you can do fragmentation. You can also use chemical methods like magnesium chloride, not so much in vogue, but you can tune the fragmentation size. So altering time exposure to DNase, altering frequency of sonication, altering strength of MgCl2. So all of this is important when you want to get your DNA library prepared to one particular size. Now what happened? This DNA fragment got DNA got fragmented, but it is not fragmented like a glass vase falling to the floor. It doesn't fragment uniformly. One fragment will have this strand stopping here, one fragment will have this strand stopping here. So you can see there are a lot of overhangs. Okay, and these overhangs are a cause of problem because these have to be repaired. All right, and what you do is you use a combination of polymerases with 5' to 3' and 3' to 5' activity. What they do is that they make these in sticky ends into blunt ends. So if this is a break that is here, it copies the strand on the opposite part, and similarly this copies the strand on the opposite part and makes a uniform sticky end. Similarly, if an overhang is there on this side, it just chops it off. Alright, so this is how the DNA is there, and it must be end repaired. Okay, so this is a step called as end repaired. Now because it is end repaired, you also do the next step, which is called as A tailing, wherein you add an extra A base. Okay, any polymerase can do this. They are often combined into one step, end repair, A tailing step. So now this is end repaired, you add an A. Now I'll tell you why we need to add this A. You need to add this A because how will your adapter get attached to it? Alright, so this adapter that is there contains a T overhang which binds to this A overhang and attaches it to it. This is not something rocket science. People who have been doing molecular biology and cloning have been doing it for hundreds of years, months or whatever, but they have been doing it for the last 20-30 years or at least, because that is how recombinant DNA technology works. It is based on cloning, and cloning requires that you have an insert which is put into a plasmid which has a T overhang and an A overhang, and the cloning takes place. So what you have here is a sequencing primer binding region, a sample index, and an instrument specific adapter here, a P5 and a P7, and this library undergoes PCR. It gets amplified and using P5 and P7 primers. So what are adapters? The term library refers to fragments of DNA containing flanking adapters. I described what are adapters. They are instrument specific stuff, sample indices, sequencing primer binding region, and inserts. Adapter help bind the template to sequencing surface. So now this is your stuff that is there, your DNA strand. Because this adapter is there, it will bind to its corresponding region on a solid substrate. In case of Thermo Fisher, this solid substrate is on a bead. In case of iron torrent, iron torrent, it's on a bead. In case of Illumina, it's on a glass slide. So your flow cell that you see basically contains a lawn of these primers. They describe it as a lawn of primers which are like this, to which the DNA strand comes and binds because of the adapter. The adapter contains sequences which are complementary to what's there on the flow cell. As Shweta described, in a patterned flow cell they are present inside wells. In an otherwise standard flow cell they are this way. Okay, why do we use index? Obviously because you don't want to sequence only one sample, you want to multiplex to get the bang for your buck. Okay, so once the sequencing, how this happens is you have first when sequencing occurs, it starts from the P5 end. This gets sequenced, the sequencing binding primer binds to it. This you don't know what happens, but the sequencing binding primer is present inside the box of the sequencer. So it has a sequencing kit inside which this primer is there. When you start doing the sequencing primer will bind to it. Then the other strand is read. Then it is read in the other way, like this. Okay, and the indexes are read. At the end of it, there are 4 fast queues: R1, R2, I1, I2. You never get to see I1, I2. There, unless you modify your sequencer and put in that special instruction that please generate I1, I2, it will not throw it out. It is important if you are going to use UMI based sequencing. Sometimes UMIs are incorporated into the indices, and for those purposes, you need to have these I1, I2 business. Now, how to prepare library? So let us see capture based library prep. Alright, in capture based library prep, what you want to do is you want to prepare, so far what we described library prep was for whole genome sequencing. Based on this, you take this, put it into sequencer, you can get whole genome, because we didn't enrich for anything. We unbiasedly added adapters, did PCR for the adapters, and then whole genome library we got. Now this whole genome library, we are not interested in everything. Remember, we are interested in our gene panel. So this WGS libraries are pooled together. The reason they are pooled together is that you want to reach a critical mass of DNA, typically 1 microgram, so as to enable capture to be successful. Okay. Now how this is done? This is done by a library pool. Okay. So this pool, that panel you gave to that oligowala now has designed a set of probes which are complementary to gene of interest. They are exactly 120 bases long and they contain biotin at one end. Okay. So this library pool now gets hybridized to these. These probes are complementary to region of interest, synthesized by IDT, twist, GenScript and whatever. You can design these yourself. Picard has a program wherein you can use to design these by yourself and order them from any oligonucleotide person, but they are expensive. If you want to order a biotinylated oligonucleotide, single one will cost you 9,000. Our panel has 3,000. 9 3s are 27. 27 lakhs it will cost you, or 30 lakhs it will cost you, but lifetime supply of oligos. तुम मर जाएगा but वो panel खत्म नहीं होगा. It is literally that much supply of oligos. So if you are a manufacturer, you want to do it, you can do it. Alright. Now you can pull this with a magnet and streptavidin bound beads, PCR amplified, to get any sequencing library. But there is a problem. All capture based things are susceptible to off target effects. Off target capture means you want to bind IDH1 exon 4, but something else only is being bound. Okay, this is known as off target capture. Off target capture is always a problem with capture based methods. So there are ways to offset capture. How do you do that? You use blockers. Blockers are substances which are sequences which are complementary to adapter and sequencing binding primer regions, and they literally block that region and they prevent anything else from binding to it. And therefore this prevents a effect known as daisy chaining, where one thing binds to another thing binds to another and causes off target capture. So we prevent off target capture by using blockers as well as COT1 DNA. COT1 DNA or salmon sperm DNA also helps in preventing off target capture. Alright, so blocker nucleotides or COT1 DNA and salmon sperm DNA, this comes and binds to it. Pros of capture based library prep. Most important advantage of this is you can scale it up immensely. You can capture an exome. You can capture all the regions of the genome that you want at unlimited scale, only if you use capture based. There is no limitation of PCR like, so many primers can't go, so many can go and so on. Most importantly, uniformity of coverage is great, and this is something that is really helpful when you want to do CNV detection. Unlike a PCR where 3000 PCRs are occurring in a multiplex manner, it is difficult to make sure everything amplifies together. But in a capture reaction, there is no amplification. There are no primers. So everything is uniform. The uniformity of coverage is great, and it also does well over GC rich regions. And it's great for calling copy number alterations. Cons: sample must be pooled. You can't just do one sample. DNA quality must be good. High quantity of DNA is required. So you can't, it doesn't do so great. It doesn't like paraffin fixed tissue and cell-free DNA. These are improving, but DNA quality generally must be good. Not forgiving of poor quality samples. You try to do this on a paraffin or you try to do it on a bone marrow trephine biopsy, and you will see what coverage you get. It will be miserable. Okay. Wait, what is this? Sorry. Alright, and off target capture must be offset. TAT is longer because of the workflow. Typically you are looking at 4 to 5 days for doing, or at least 3 to 4 days for doing library prep. It is painful. Amplicon based library prep. Okay, so in Amplicon based library prep, you have DNA or you have cDNA, reverse transcribe, straight away do PCR with primers. Once you do the PCR, what you want to do is your PCR product is formed. The PCR product is then less than 150 bases. Why it is less than 150 bases? You have designed the primers accordingly. This is how Thermo Fisher makes all their money. Alright, Thermo Fisher's everything is Amplicon based. They don't do well with capture based stuff at all. Everything is Amplicon based. So what is proprietary to Thermo Fisher is that once these PCR adapters are formed and you get this Amplicon, you can remove the primer sequences using something called as a FUPA reagent. That FUPA reagent is basically allows you to digest these primer sequences, and then you can attach adapters. You can do this yourself, but it is you're stepping on their patent. You need to be taking care of that. So once you ligate their adapters, you can amplify with sequencing primers, and you have a sequencing ready fragment. Like I said, FUPA enzyme is proprietary, which is used to digest sequences, and they have a patent for it. Pros and cons of library based prep. No need to pool samples. Can do single samples. I get one sample, I do it, finished, end of story. Okay. Forgiving of poor quality samples. Low quality DNA, no problem. FFP DNA, no problem. Off target capture, negligible. TAT is short because of the workflow. Cons, allelic skew, and this is true for any primer based workflow in the world. Anybody says I don't have allelic skew is, bluffing. So allelic skew means one region of the genome gets 1000x captured, other gets 300x captured, gets sequenced. Why? Because that region is not GC rich, and this region is very GC rich. So this is going to be less uniformly amplified. The other thing is more uniformly or more effectively amplified. As a direct disadvantage, copy number is not great. Imagine one region is 1000x coverage, one is 300x coverage. By default, that 300 wala is a deletion. No, but it is not the case. It is because of amplicon inefficiency. Fusion calling is not possible for unknown partners. So only if you know the other sequence, you can add to it. If if one partner is unknown, the forward primer will bind to what? It cannot work. Alright, so poor coverage over GC rich regions, this we all know. There are certain regions of the genome you need to be aware. You can do something called as increased tiling, that means design more number of amplicons for that. Typically C, BPA, SRSF2, U2AF1, these are all regular suspects where you will get high GC rich, and these regions will cause you trouble. Go back to your assay and take a look. Yes, you will find कि अरे ये तो problem ही देता है. Third way is molecular inversion probes. So molecular inversion probes were invented by J. Shendure and George Church. J. Shendure described this technology in his paper way back in 2009. I don't know what I was doing in 2009. In 2009, he described this technology to capture all the coding regions of the genome and called it exome. Today, every kid who's in genomics will tell you what an exome is. So the first exome was done using molecular inversion probe technology. You can use lot of these MIPs. These MIPs are bind to regions complementary to what you are trying to capture. They copy this region to create this reaction called as gap fill and ligate to create a circle. This undergoes rolling circle amplification to get a sequencing ready library. Okay, this is still not commercially available in India. Some companies have made it commercially available abroad, but not so much. Just to show you an example, we initially started a lab in 2016 using MIPS. So we get this whole 1200 capture oligos of MIPS. Each contains 300 or 400 microliters of one oligo. All right. So what you do is you take from this just 2.5 microliters, pool it into 10 pools. From this you take 2 to 4 microliters to form 4 pools, and just take 10 microliters, which is enough to sequence 200 patients. So you can see that in an upfront cost of a few lakhs, you can get a lifetime supply of NGS oligos. Okay, so this is a very cheap and effective way of doing library prep. Very cheap, very well suited for small NGS panels. One-time synthesis will last for thousands of NGS libraries. Cons, it's still not perfect. It doesn't do well with GC rich regions. C, BPF, forget about it. Poor performance over low quality DNA, forget about it working on FFP, cell-free DNA, and so on. Uniformity of coverage is not great as compared to capture and can't detect fusions. So if you can see this is a coverage data of this. You can see some regions are being sequenced very well, some regions are not so very well, and some very small percentage, 1% or so are very phaltu. So this is typical of MIPS. Fourth is anchored multiplex PCR. So this is also a way of doing sequencing. Now this paper came from John Efrate's lab in Harvard. All right. So what this came in 2014. What you do is take dsDNA or cDNA. You fragment and repair 8 tail a half functional adapter. So one adapter is there, but only one side contains a P5 or P7, whatever is there. And then once it binds to it, you do amplification using a gene specific primer and a universal primer for one end to get a sequencing ready library. You can add ion torrent or Illumina adapters. And the biggest advantage of this is you can detect fusions only when you know one partner. So if you want to detect a ROS fusion, you don't need to know the other partner. You can just by sequencing ROS, you can find any fusion partner it has. So they showed this, that you can detect fusions and so on. This company has a very funny history. So this company spun out from Harvard to be called Archer DX. Okay, Archer DX got sold to Invitae. Invitae then got bankrupt. It got then purchased by IDT. So now IDT sells these products. Then at the same time, I think QIAGEN post-COVID became so much money, they said we can do whatever we want. And they essentially copied this assay blindly and totally. And the Archer sued them. They said, okay, take some money, keep quiet. And they gave them some money because they had so much money to give. And they essentially copied it, and it's called as QIAGEN's single primer extension approach. Same thing. You have a one-sided adapter here which binds to this, and you do amplification using universal primer. At that point, I wanted to use Archer's assay, and I said, how much it costs? They said 26,000. I was like, I also can't afford it, let alone patients. So at that point we said, let us make our own thing. So we made our own thing, which was which is an assay called Narasimha, and same thing, fragmentation, end repair, A-tailing, add a spume sort of substance here, which contains universal primer binding region as well as a UMI, and then you can amplify using a whole set of these genes. So you can identify any fusion that is there, two modules, whether lymphoid and myeloid. At that point, we showed in a proof of concept works now more than 4 years we have used it. Current version has undergone several changes, and this is the final version that is there. Now this is a patented approach. We call it Aloha-seq, and same concept. All right. So pros and cons of this is no need to pool samples. You can do single sample at a time. Forgiving of poor quality samples, low quality DNA, FFP, off target capture is negligible, TAT is short, can detect fusions irrespective of knowledge of other partner. Cons, allelic skew, unequal coverage, copy number alterations is not great at all. So same problem with amplification based approaches. GC rich coverage may not be great. You can aggressively tile primers here to offset it, but still it is not as great as capture based methods. There is also error corrected sequencing. I'll talk about it when we do that talk all together. So quality control for NGS, setup of NGS laboratory should follow general principles. Strict segregation of PCR pre-setup, post areas. Once WGS library is made, capture needs to be done in post PCR area. There is a separate kind of contamination which people don't talk about. It's called bioinformatic contamination. If you are using indices and you add one index drop also into another thing, you are giving prasad to entire run. So that one fellow's NPM1 now goes distributed in everything. Alright, so you have to be very careful. Even though there may not be a sample there, even if index goes from here to there, you are adding contamination. So this is a unique thing to genomics you must be aware of, especially if you do MRD testing. If your cutoff is 5%, doesn't matter. But if you are doing MRD testing, it has serious implications. Quality assessment of library must be done prior to sequencing. I think I'm stepping on many people's talks here, but you need to do it. All right. Check tape station, adapter contamination, quality assessment of libraries are very important. You need to look at coverage. Is the coverage adequate? What is coverage? So how are we doing on time, Miss Neha? Just 2 minutes? Okay, very good. So I better hurry up. So coverage is a number of aligned reads that contain a given nucleotide that are there at a nucleotide position. Uniformity of coverage is whether the there is variability in coverage. I just showed that by that plot on those MIPS, that coverage may not always be uniform. How much coverage? So this is everybody's favorite question. How much coverage? Basically they want to know how to save money, Baba. This is very expensive. So correct, for germline or Mendelian disorder testing, minimum 30x, ideally minimum 50x. Okay, sequencing is becoming cheaper. For somatic, ideally 1000x, but 500x के नीचे लेना ही नहीं है. Okay, you are going to miss signaling pathway mutations, RAS, PP53. In solid tumors, you also need to know about what is germline, what is normal tissue contamination. All right, so tumor fraction, because that also will affect coverage. Coverage on target means what you want got sequenced. Off target, what you want didn't get sequenced, something else only got sequenced. Near target or off target is something you see all the time in capture, because you are not able to control how fragmentation goes. You will get bajuwala also. Okay, little bit of nearby areas, it's okay, it doesn't make any difference. Then total read number, mapped reads, on target, all these quality metrics are something you will need to generate using bioinformatic tools. Breadth of coverage, how much region of the genome got covered. Depth of coverage, how many times it got covered. Okay, so these are two things you need to be aware. Just like you have cell controls, you have controls in CBC counter. This is an MRD assay where you have NPM1 high precision and low precision control, 0.02 and 0.2% VAF. We published this some time ago. You can run these controls to see how your assay is performing over time. So no, NGS is not always 100% successful. Depending on the type of mutation that you want, it can be missed. So this is with MIPS, where CalR 52 base pair deletion is simply missed. Today we have become much better at detecting MIP indels, especially with newer bioinformatic tools and algorithms, even with FLT3. But a notorious problem here is FLT3. Okay, FLT3 large ITDs, you have to run orthogonal assay, you have to run FLT3 ITD with fragment length analysis. Anybody in world tells me my FLT3 is perfect, I will have side conversation with him. It doesn't work. Okay, so MLL PTD, PAX5 ITD, sometimes CalR, sometimes large BRCA gene mutations. If you have thought of labs, अरे वो BRCA करो लेकिन MLPA भी करो, क्यों double पैसा क्यों? Because it is not perfect. Because large deletions are a problem with NGS assays and you are going to miss them. GC rich regions you need to be aware. Then some mutations are not always, but can be seen below the limit of detection of an assay. This is typical of JAK2. Alright, JAK2 mutations can be at 2%, 3%. You will miss them because your assay is typically set up at 3 to 5%. Always for a MPN patient, run this on the side. A real-time PCR assay. So understand clinical context. If an MDS patient is there and the sample given to you is extremely dilute, some genius sent peripheral blood for MDS and corona NGS because he forgot basically to send bone marrow sample when it was being drawn. So you are going to get negative assay. You are sequencing lymphocytes. So this common sense has to be there. You need to understand pathology context as well. Okay. So of course the usual thing of accuracy, precision, sensitivity, specificity, all of these matters. I would refer you to a book by Kulkarni and Pfeiffer, Clinical Genomics, which defines this. Anybody doesn't have book, please send me a mail. I can share a PDF copy with you. Alright, so please don't share that. Alright, so NGS testing processes and their potential workflows at different errors, stages you can get errors. Once again I am stepping on someone's shoes, I am 100% sure, because somebody else is supposed to give this talk. With that I am saying thank you and happy to answer any questions you have. No question. Rajiv.
So Nikhil, so when you talk nicely about how to select your panels and design. So when you try to design a new panel, so do you also try to look for the ethnicity and the local data which is available? And because especially in the solid tumors, there is very limited information available from the Indian data set, and then we have a confusion whether we should always replicate what other people are doing, or should we look into that our own?
This is difficult question to answer. My experience in this is that there is not so much of ethnic bias, especially at least in blood cancers what I've come across. when they say India में ये mutation ज़्यादा दिखता है और say on, the experience of the samples is less. When you look at large cohort of samples from India, you will find most of the things match with the TCGA or gold standard data. It generally matches. There may be age bias, there may be hospital referral bias, but it's not there. If such data is there, by all means, read the literature thoroughly. If such is there, please add it to your panel. But in my experience, in hematology at least, we have not found such a ethnicity associated bias. Having said that, in solid tumors, there are some India specific cancers which may not be well represented in TCGA atlas, such as your gallbladder cancer, such as nasopharyngeal cancers, such as things that are specific to the Ganges belt or the northeast of India. So those things, yes, by all means, it is more difficult there. So
available mutations, right? So basically it's a hotspot panel.
Largely, yes. So hotspot का मतलब होता है IDH1 exon 4. But most of the genes that we have in our panel will take a look at the entire gene.
Okay.
Only few things we look at hotspot. Mostly we try to sequence everything,
So
the whole gene.
How to decide upon that, sir? Like
How?
How to decide upon that? Like for IDH1,
From literature. Literature and experience. Now we have been seeing since IDH1 was discovered in 2009, right? 2009 seminal paper by Tim Lay. We have not seen anything else come across. So keep a hawk's eye out for literature, and you know that something is there which comes again and again for IDH1 is the same thing. So it's not going to change. But there are some genes like KIT. Let us give the example of KIT. Exon 8 and 17 everyone knows. But do you know that there are mutations outside of these which happen all the time? There are exon 16 mutations, there are exon 20 mutations, there are exon 12 mutations. So for KIT we have taken everything. So how to note that is when you go to Cosmic, you will see this. You will see that there are mutations happening everywhere. They are rare, but they are there. So you have to take those.
So and then when we go for the NCCN and all,
When we go for
NCCN, and then for each disease, like for MDS and AML, they give sets of genes to be looked upon. Can we directly go for those genes and decide our panel accordingly, sir?
So no, this is so what we are pathologists. For physicians who want to risk stratify, essentially NCCN is a risk stratification tool. You want to fit patient into low risk category, treat with chemotherapy, high risk category, treat upfront with transplant, and so on. But as pathologists, these do much more than risk stratify. They help you diagnose diseases, they help you classify diseases. We see this all the time. Patients with confusing diagnosis. You're not reaching only any conclusion after doing flow and morphology. This will help you reach a diagnosis, because then it falls into that. So you have to take account all of that as well. Don't go by NCCN. Go by disease biology, because it will help you diagnose that disease. Yes.
Is there any cutoff for the near target region? This much percentage should be there. Off target, if it is like lesser than 0.5%.
Yeah. So if it drops below 60%, this is just experience. There's no hard and fast rule. Say you are essentially sequencing something else. So if it drops to below that 50% or so, just repeat your library prep. It's unfortunate, but you have to do it.
And then fold 80 base penalty. Fold 80 base penalty.
Oh, I am not familiar with that. Please educate me.
So, like in the exon sequencing, when we look at the twist or Agilent, they say like the fold 80 base penalty is close to around 1.2. Ideally, it should be around 1. If it is around 1.2 or 1.4 in the 6 GB or 8 GB of the data, we are getting everything we want. But in the real life world situation, in our laboratory setup, we are always getting fold 80 base penalty above 1.6. Like we have tried some 4 different exome kits. All the 4 different exome kits, this is what we are getting it. So I just want
no idea. So we have not come across this metric. Maybe it is specific to Agilent. I'm not sure. We use for exome, we use Chiastic. They don't seem to have this metric, but I will look into it. I am myself not sure. Debasita.
Also, sometimes you are kind of twigging with some CNVs, like some chromosome 5, 7, whatever 8. So in that thing, so how you are doing that? Again for that chromosome part, maybe I need.
So that is a separate design. So I did not speak on CNV panel design. That is a separate consideration on how to do that altogether. But I can tell you. So well, I wait for the lecture, no, a whole CNV thing, because there is a lot of thing on how SNP arrays were done, what were done when they used to do SNP arrays, and based on that SNP arrays, we have designed those chromosome backbone. So it is basically like that. I think our fellows will tell, but typically 10,000x bulk and at least 2,000x 2 or 3,000x UMI based, correct? MRD depth what we go? Panel? Ballpark, no? 10,000x bulk and 3,000x UMI. Oh, it's a expensive essay.
Yeah, very interesting talk, sir. Just have a question. If we have to make a panel for non-cancerous, like inherited disorders, do we have a website like CBI portal or?
Yes, of course. So for germline disorders, there is HGMD database. There are databases for sure that will be available. So germline disorders, no, there are databases available which are disease specific. So you need to do that thing. So for example, inherited bone marrow disorders, there is a separate website that is maintained by St. Jude's. Some researchers in St. Jude's have a SAMD9, SAMDL9 database. Somebody else has something. You have to do, extra work to get that generally. But databases are there.
Thank you.
UCSC and ClinVar will also have.
Correct. As Gaurav said, UCSC, ClinVar will also have these things. Hi.
As you told, sir, that there should be like in case of lymphoma, you went later on you reached to 150 genes only. So what is the superiority of NGS over MLPL? Even in MLP we can go for 200 gene.
So simple thing is MLPA is one additional test, no? NGS in a single test you can do CNVs also, you can do mutation detection also, you can do fusion also. MLPA means you have to do NGS. In any case you need to know what mutations, indels, windels are there. Plus you need to do MLPA to detect those deletions.
And whenever you are suspecting deletion like in alpha thal, then MLPA.
Because so you need to be aware of disease context. Like alpha thal will typically have those alpha, the 3.2 and at 4.2, they are large deletions. So you need to design your assay in such a way that it spans that entire alpha globin gene, or you need to do long read sequencing, or you need to do something else to make sure those are met.
In addition to, like, I want to answer the ma'am's question, like in thalassemia again, Ethernet is a different website they are doing. Like ACMG guide, as per they are classifying it as per the ACMG guidelines.
Yeah, thalassemia is one area where they love their old nomenclature and they refuse to leave it, right? Okay, thank you.
Thank you, sir, for this enlightening talk. Your insights have certainly helped everyone understand the intricacies of panel designing and NGS library prep. Next up, Mr. Prasanna will talk us through the organization and infrastructure required for setting up an NGS laboratory. Please join me in welcoming him.
Good afternoon everyone. My name is Prasanna Bhanshe and I'm going to talk on organization and infrastructure needed for NGS laboratory. So setting up a next generation sequencing lab is an important and complex process and plays crucial role in modern molecular biology and genomics and personalized medicine. So the importance of the NGS laboratory. So NGS laboratory play a crucial role in field of genomics, revolutionizing how we understand and analyze the genetic informations. it also helps in foundational in genomic landscape and supporting wide array of application from basic research to clinical diagnostic and personalized medicines. Their ability to produce detailed and large-scale genetic data with speed and precision has transformed our understanding of genomic and its role in health and diseases. So organizations of NGS laboratory, the setting up a next generation sequencing laboratory requires careful planning and organizations to ensure that you have a right infrastructures and equipment and procedure in place. So my presentations has divided into 5 major sections, starting with the planning phase of the laboratory, equipment and technology acquisitions. We need to understand good laboratory practices in genome, staff training and staffing and training, and most importantly the quality management system in a genome laboratory. So in a planning phase, evaluate the physical space needed for equipment, workstations and storage, ensuring compliance of safety and regulations, and also facilitating efficient workflow in molecular genome processes. Identify necessary utilities and infrastructures needed for a genome laboratory, like water supplies, electricity, ventilation systems, along with the specialized infrastructures like PCR hoods and biosafety cabinets. Also assess the qualification and training needed for a laboratory staff, including their ongoing operational expenses. Estimate cost associated with the purchasing essential equipment, which is a major chunk of the cost is involved in this section. Laboratory supplies and facility modification. It also ensuring comprehensive budget that cover all these sections of the laboratory. Explore some potential funding opportunities from government agencies or private fundings. So set up a system that also required for biological, chemical and sharp waste. So follow the local regulations, like in Maharashtra we have MPCB, that is Maharashtra Pollution Control Board, so we have to take the permission from them. Now the equipment and the technology acquisitions, so acquiring right equipment and technology for next generation sequencing is a crucial and in terms of development, and it is directly impact the quality and speed and reliability of the results. So we going to see this in detail. So outsourcing strategy. Now establish the clearer criteria for evaluating the potential suppliers, including their reliability, quality of products, compliance with the industry standards, and ability to meet the delivery timelines. Conduct thorough cost benefit analysis for outsourcing decisions, considering not only the purchase price but also see the long-term maintenance cost, warranty options, and potential discount for bulk purchasing and optimal budget allocations. It is also important to develop a strategic partnership with the key suppliers and manufacturers to enhance the procurement efficiently, to gain access to the technology, and negotiate favorable terms can support the laboratory for ongoing operational speed. So we do in our setup, we have the reagent rental contract with the premas, so kind of thing you can do also. So you don't need to purchase the instrument, so you have to purchase the reagents and they provide the instrument. So it's a win-win situations for both of them. Now this is a major part of the NGS laboratory setup, which is essential equipment for molecular genomics. So depending on your need and infrastructure, the next generation sequencer, which includes Illumina, Thermo Fisher, Aviti and other NGS platform can be considered. So these are the high throughput instruments, sequencing instruments that are crucial for comprehensive genome analysis, which allowing for rapid sequencing of an entire genome and provide vast amount of data for research and the clinical considerations. For a laboratory, the high throughput laboratories, so liquid handling system may help you to get the library preparations and primer allocation, sorry, the reagent allocations. It is also important to have the tape stations and qubit 4 which is required for the library quantifications and quality check. For the thermal cycler for NGS preparations and PCR is required, so you have to have this thermal cycler in your setup. In case of high humidity in the NGS room, you have to analyze that. If case the humidity is more than 60%, then you have to have dehumidifier in your setup. So these are the some sample preparations. Sample preparation equipment start from micropipette, mini centrifuge. So these are the list of the instruments required in the NGS laboratory. So these are the some specialized equipments for NGS laboratory. So they start from Illumina NextSeq 1000, 2000, NovaSeq, MiniSeq. Then we have AVIT. These are the also advanced instrument like AVIT from Element Biosciences, Ion Torrent from Thermo, then next Oxford Nanopore and PacBio. So these are the specialized instrument required in your NGS laboratory. Liquid handling system also plays important role. As I told you, it can save your time. So investment in this freezing equipment is also important to store the sample and the reagents. So invest in -20, -80 and -180 degree storage which required power backup supplies. Use laminar flow hoods and biosafety cabinets to maintain contaminant-free environment. High performance computing system is also required for CVS for the data analysis, for the storage solution of large data sets. Also important to have laboratory information management system. It may be for in-house developed, but it is very helpful in arranging the reagents and the sample tracking. The third section is the installation, calibration and maintenance of the instrument. So the installation qualifications is must be checked. So installing and calibrating NGS next generation sequencing instrument is a critical process to ensure accurate and reliable sequencing data. Ensure that lab space meets the infrastructure and instrument dimensions, ventilations and power requirements. So check that your laboratory have adequate environment like temperature, humidity, free and cleanliness, so that most of the NGS instrument require a dust-free environment. So we have to check that also. For the instrument with the optical, so for the calibrations, instrument calibration is also required. So optical calibration, temperature calibration, reagent delivery calibration, and flow cell calibration. So this has to be done after the installation, immediately after the installation of the instrument. Also, we have to validate the performance validations for the run control samples and base calling accuracy, library quality checks, and cluster density. This is applicable for Illumina sequencer. Apart from that, the documentations record also required to follow the record of calibration data, error logs, validations run, and troubleshooting has to be documented, and it has to be conveyed to the respective concerned person. Now the maintenance part, the preventive maintenance also required on daily basis. So depends upon the manufacturer recommendations. The preventive maintenance has to be done daily, weekly, monthly, or annually or biannually. So cleaning protocols also, the instrument cleaning protocol has to be scheduled as per the manufacturer requirement for optics, thermocycler and reagents compartment. Also need to see the software updates which is required to fix the bugs, improve the performance of the instruments, and new functionality in the instrument. Now, the good laboratory practices are crucial in genomics laboratory for several reasons. These practices ensure the accuracy, reliability, and reproducibility of the experiment, which is especially important in dealing with the sensitive clinical data. So, designing the ideal laboratory, the space need to be considered, and the area has to be defined logically. So this is a layout on workflow of laboratory. Start from sample reception, so where you can receive the sample for next generation sequencing. You can use the laboratory ID for the patients, and you can enter this data in your database with the help of LIMS, which is a laboratory management system. Now the other area is the pre-PCR room, which is especially made for nucleic acid isolation from of DNA and RNA. It also required to have dedicated area for reagents and master mix preparations and template additions. So we have this separate area, like for example for the RNA and cDNA separate additions, we have different hood, and for the DNA addition we have separate area. So that has to maintain to avoid the contamination. The another area is the NGS laboratory preparations area, where you can prepare the laboratory, prepare the NGS library, where you can follow the process like fragmentation and repair ATL, adapter ligations, and amplification. So the amplifications area, the post PCR area, and gel electrophoresis area has to have different region or area, otherwise there is an issue of the contaminations. Then the next generation sequencing room, it is a minimum, it is an area where you have to minimum access to this instrument. Very few and trained persons can attend this area, otherwise there is a issue of a contamination, as well as a issue in the Now next is the data analysis in the reporting room. The data analysis area is used for performing bioinformatics and various softwares and high servers. Now this is a special area required, and it is a and it is very important for large data set which is generated by the next generation sequencing. There is another area where archival and storage room is also required, because we as of now we don't have the dry area for the storage. So that area is also important, because most of the boxes is lying in outside the laboratory, so it's not look good, and it has to have some separate area to store the tips and plastic waste. So these are the general the decontamination approaches for molecular laboratory. So use of positive displacement pipettes, disposable filter tips, use of sterilized single-use plastic waste, also required to minimize the risks of amplicon carryover on clothing, hair and skin. So hairnet and dedicated septic glasses need to be used. Use of nucleus free or autoclave water, aliquots, nucleotides and multiple freeze thaw will cause degradation, that has to be considered. Wipe test or swab test can be implemented regularly. Also required to have a clean work area and equipment routinely, clean PCR workstation at the start and end of the work day or per run. At least you have to do the 70% ethanol cleaning and 10% sodium hypochlorite and DNA away. Clean the equipment and clean the door knobs and handles the freezers. So these are the general decontamination approaches for the molecular laboratory. Now the reagents and consumables, the single entrance reagents are used for the amplification, should not expose to the other areas. So high quality critical reagents has to be securely supply chains for the laboratory preparations, sequencing reagents, enzymes, probes and other reagents necessary for the sequencing has to be maintained properly. Consumables like stock pipette tips, micro centrifuge tubes, PCR, these are plastic waste, has to be maintained properly. Area-wise reagent distribution, so arrange the reagents as per the area-wise, like the pre-PCR area has the set of reagents, PCR area has sets of reagents, post-PCR and sequencing reagents have different storage place. So to maintain the inventory management, so the molecular inventory management is very critical. The molecular stuffs are in a large number. There are a lot of reagents, primers, plastic waste. So we have to have this management system and inventory in place. So for inventory management in a molecular laboratory is critical for ensuring smooth operation of the experiments, minimizing waste, optimizing the cost is also important. So to get the effective inventory management helps to maintain accurate record of the supplies. It prevents the shortage and overstocking. And to maintain this management system, we have the best practices for the inventory management in molecular laboratories, which can create a comprehensive inventory list, set a reorder points, regular audits and stock taking, training and access control, documentation for the compliance. So these are the some of the best practices mentioned in this presentation, and these are the we have in-house inventory management system. We have the software, so we use this inventory management system for our molecular daily basis routine. These are the safety compliances. So it's the general safety compliance has to follow, start from eye wash stations, showers, protective equipment which is designed and dedicated area for PPE storage, gloves, lab coats and safety goggles. There has to be use proper storage of cabinets for chemicals and reagents. Emergency exit and first aid kits which required for accessible locations, autoclave stations. So these are the general considerations, safety compliances, and of course the biomedical waste management. So identifying required skill set is also required in staffing and training. So technical experts in genomics are required for effective conduction of the experiment and analyzing genomic data. So before selecting, you have to understand your approach and the importance of the laboratory section. So if you require the wet lab experience or the dry lab experience. So understanding the person who employed need to understand the library preparation and the sequencing workflow. For a dry lab experiment, experience is required to have a bioinformatics experience, and the continual training program and education is required in terms of workshops, seminars and online courses. Now the quality management system, the preparations of SOP, it helps the organization to run effectively and consistently. So SOP helps to ensure that everyone in an organization follow the same processes and which can be improve quality of work. Proficiency testing and ECAS, it gives the benchmarking. The labs can compare their performances against a large number of peer groups. Quality improvements, it provides valuable feedback on where the labs may have weaknesses in their processes. And the compliances in many regulatory bodies require participation in ECAS to maintain the certification and accreditation. In enable audit and quality indicator, the measurement of monitored and record of quality indicator as a part of quality management system which is required for the NABL. Start with the TAT, percentage of run failures you can monitor, and monitor frequency and acceptable ranges of your assay. So key takeaways of this establishing an NGS laboratory. So good infrastructure ensure the data quality and minimizing error due to the contaminations. Plan for scalable workflow to accommodate future growth and new technologies. Build a relationship with the external institutions, suppliers and research partners to stay competitive and innovative. Reliable data storage and management system to handle large scale NGS data. Adequate computational power and bioinformatics supports for the data analysis. It also continuously adapt to advancement in NGS platforms and bioinformatics to remain at the forefront of the genomic research, and ensure a strong leadership and continuous staff training to for evolving NGS technology. These are the references. Thank you very much.
Thank you, sir, for your informative talk. I'm sure your talk provided invaluable guidance to everyone here and how to effectively establish their own lab. Now, Ms. Pratiksha will provide us with an overview of the workflow involved.
Good afternoon, everyone. So in this presentation, I'm just going to give you an overview of what all things that go in a genomic laboratory or a molecular laboratory. So the overview will consist of 4 sections, that is sample processing, library preparation, sequencing, and the bioinformatics. So in a molecular laboratory, the area segregation is of utmost importance. So usually a molecular laboratory will be divided into pre-PCR, PCR and post-PCR, and there has to be a unidirectional flow, meaning stuff from PCR cannot be used in PCR area, and similarly stuff from post-PCR cannot be used in PCR or pre-PCR area, because this is going to lead to crossover contamination. So let's see what stuff happens in a pre-PCR area. So here we usually process the sample. So types of samples that we receive in our laboratory. So as our laboratory deals majorly with hematolymphoid malignancies, so our major load of the samples are peripheral blood and bone marrow samples. But we also in a minor quantity, we also receive CSF samples, saliva, FNSE sample, blood and bone marrow smear slides, and sometimes biopsy tissue blocks as well. So there has to be some pre-analytical checks that we have to take into consideration before receiving the sample. So the sample has to be collected into K2 EDTA vacutainers, because EDTA, as we know, is an anticoagulant and it will prevent clotting of the samples. Now the sample transportation has to be maintained in a cold chain, because most of our tests are RNA based testing. So the logistics of the samples have to be carried out at 4 degrees temperature, and temperature monitoring has to be done. So when you receive the sample, there has to be a cross-checking between primary and secondary labeling. No mislabeling samples have to be received. Then next is the sample quantity and quality. So obviously we we reject the samples that are clotted and hemolyzed, and the volume of the sample that we accept is for peripheral blood usually 7 to 10 ml, and for bone marrow it is 9 to 10. Now, what is exactly sample processing? So sample processing is RBC lysis and further DNA and RNA extraction. So RBC lysis, as we are interested in WBCs obviously, because those are the nucleated cells and we are going to obtain the genetic material out of them. So we are only interested in WBC pellet. So RBCs also have a heme component which is going to cause hindrance in the downstream process of PCR. So RBC lysis that we do in our laboratory is using isotonic ammonium chloride solution, and along with EDTA and sodium bicarbonate as adjuvants, we carry out lysis of the RBCs, and what we get is an intact WBC pellet. So storage of those WBC pellet is of utmost importance. So what we use is RNA later, which is a RNA protectant, and it basically will stop the activity of RNase enzyme. And those pellets are then stored at -80 degrees. And so making of pellets is important because number of tests can be performed at depending on the diagnosis, and storage also will make sure that your RNA is in good condition. So also very important is that the processing of baseline and MRD samples have to be done separately, because MRD samples, as we know, are we are looking for a molecular so we are looking for MRD samples. We do not want any crossover contamination of the baseline sample into the MRD samples. So as you can see in the picture, in the first, so the blood or the sample goes through a series of buffer washes, and what we get in the end is a WBC pellet, and those WBC pellets are then submerged with RNA later and stored in -80 degrees. So now after your WBC pellets are prepared, we will go for nucleic acid extraction, so RNA and DNA extraction. So trizol, which is a conventional method for RNA extraction, was to be used before, but now nobody uses it because of suboptimal RNA quality. Spin-based column and magnetic bead technology is what are people using right now. Spin column based is also a good technique, but sometimes what happens is the limitation of spin column based is those silica membrane, they get clogged if you are using too much of sample. So magnetic bead based technology by Promega is what we are using, and in our laboratory. And for similarly for DNA extraction also, there are a lot of other methods available in the market, but we are going with magnetic bead based. So the nucleic acid extraction by magnetic beads is nothing but use of paramagnetic beads, which are positively charged and onto which your negatively charged genetic material binds, and later can be separated from the supernatant by applying a magnetic force to the outside of your tube. So so these cartridges are ready cartridges with reagents. All you have to do is prepare the lysate, load it into the sample well, and you can just simply load those cartridges in the machine and on the machine. So as you can see, so lysate preparation for RNA is those WBC pellet. First you have to homogenize the pellet, and then you have to add a lysis buffer into it and make the lysate. For DNA extraction, you just simply have to remove the RNA later, add your PBS and homogenize the sample, and you have to add it into the first well of the cartridge. And as you can see in the picture, the sample gets mixed with the magnetic beads, and it goes through a series of washing, and at the end of it, the nucleic acid is eluted. Next is quantification. So in NGS laboratory, your sure shot quantification technique is to use a qubit, which is a fluorimeter. Nanodrop is generally not recommended for quantification as it is not reliable, but you can use it for protein contamination in the sample. Now the next area is PCR and post-PCR. So when a sample is received in the laboratory with NGS requisition, so first the clinical findings of the sample have to be screened. Later flow and morpho findings, and along with cytogenetic findings are to be screened. So for example, if the morphology is consistent with acute leukemia or myelodysplasia, the first thing we will do is the conventional PCR with for NPN1, FLT3 and SEPA. Why conventional PCR? Because they are quick and a basic prelim diagnosis can be conveyed to the clinician. Similarly, if it is NPN case, we will go for JAK2 and CALR conventionally, and at the end of it, all the samples go for acute leukemia panel. Similarly, for myeloma and lymphoma, separate panels of myeloma panel and lymphoma panels can be used. So in cytogenetics, if the sample test negative for common fusions by PCR and cytogenetics, RNA sequencing is carried out. Similarly, if it is a known case of CML in a blast crisis, we will go for kinase domain mutations. And if it is a known case of acute leukemia, we also do MRD monitoring. So this is the NGS panel list for our laboratory. The test marked in red are DNA based, and the test marked in green are RNA based testing. So the NGS processing for acute leukemia panel and RNA sequencing are two different things. So acute leukemia panel will use DNA as its starting material, and like we saw it in the previous presentations, whole genome sequencing library preparation happens, and we pool 26 samples. For one pool we take 26 samples, which goes for hybrid capture, and then later on the post capture cleanup takes place, wherein we remove all the unbound probes, and then a final pooling of 2 nanomole library takes place, and it goes for sequencing. Similarly, for now RNA sequencing, first we'll enrich the target by using a gene-specific PCR, and again it will go for WGS library prep, pooling of samples, and final pooling of 2 nanomole library and sequencing. Now bioinformatics. Now once the sequencing run is over, the sequencer will convert your TIFF files into CISF files and base calling files. And these files are then the sequencer, we can demultiplex them. So why there is need to demultiplex? Because we are going to load a pool of samples. So the library will consist of sample plus your indices, that is sample specific indices, which are going to get separated, and the separation of the reads on the basis of the using bioinformatics can be done. And then later on FASTQ files are generated, which will go through this series of customized pipeline, and we will generate a CSV file on the basis of which the reporting is done. So this is the basic overview, which you are going to get into detail in other presentations and wet lab workshop as well.
Thank you, Pratiksha, for the overview of the workflow. It truly helped understand the process better. Now, with this, we end the lecture sessions for day 1. Does anybody have any questions regarding the first day or the sessions of the lectures that we had so far?
RNA later, how long do you store the sample? How long you can store the samples in for RNA later if you want to extract it later?
Up to 5 years.
5 years.
Yeah.
It stays.
Yeah, it stays.
You also cryopreserve.
Okay.
So you cryopreserve the cell, or
Sir.
Pellet means RNA later में homogenized होता है ना?
After you lyse the RBCs and make the pellet and put it in RNA later and store it. Okay, how long, after how many days you extract it, like, or it can stay for 5 years like that?
That's what she said, no? It stays 5 years like that, yeah?
Okay, you can extract after 5 years also.
Yeah, it stays pretty. Triazole the people have used for many years. Basically, RNA later is some derivative of a chiotropic agent. It stays substantially. Triazole people use for many years. It's not a problem. Only thing is that your -80 should not suffer power. Our main problem is -80 will die before your sample will go. Your -80 breaks down, samples have to be removed, they have to be again shifted to new -80. All these things in India are a problem. Power cut happens, your servicing is something will happen. Suddenly they'll tell you, "Arey, you know, newer lab spaces come, you go there." So these are the main issues rather than the triazole itself.
there was one slide where you said about the samples. So peripheral blood was mentioned as 7 to 9 ml and bone marrow was mentioned at 9 to 11 ml or something. So why bone marrow was the amount? Yeah. the amount of bone marrow written over there was, I suppose, was higher as compared to the peripheral blood.
Must be type.
Huh?
Must be a type.
Okay.
No one will give 11 ml bone marrow. We get 1 to 2 tubes, to about 2 ml, 2.5 ml is what you.
And the workflow shows WGS, that is whole genome sequencing. So every case you go for.
Every case we go for WGS. I showed you in library prep. No, first step of capture is WGS.
Okay.
You have to prepare whole genome. Then those whole genome are 150 base pair fragments. You use baits to pull out from that the genes of interest that you want, and then you further do a set of library prep to amplify it.
Okay, so I now invite you all to move to the cafeteria for the lunch. Also, after lunch at 2 p.m. we'll be dividing into 2 batches. Please refer to the group indicated in your IDs. Group A and B will be in batch 1 and group C and D will be in batch 2. And for the wet lab sessions, batch 1 will be focusing on whole genome sequencing library preparation, while batch 2 will begin with bioinformatics, covering topics such as BASH, quality control and pre-processing. There are volunteers available to guide you to the locations for the next sessions. Thank you.